Dear students, welcome to PUC second year biology class. Today, let us see the chromosomal theory of inheritance. This is the topic that we are going to study in the lesson called Principles of Inheritance and Variation. In this chromosomal theory of inheritance is being proposed by two scientists called Walter Sutton and Theodore Boveri. The year 1902. These were the two scientists who have proposed the chromosomal theory of inheritance. It states that the pairing and separation of pair of chromosomes lead to segregation of pair of factor they carried. That means pairing and separation of pair of chromosomes lead to segregation. They get separated, and that will be we are separating the characters, that is factors that they carry. Sutton united chromosomal segregations with Mendelian principles and called it the chromosomal theory of inheritance. He connected the Mendelian principles and the experiment that he has conducted together. He called it as chromosomal theory of inheritance. Let us see what is the importance of that. Important statements of the chromosomal theory. Chromosomes are vehicles of heredity. One of the important statement that is being made by the Sutton and Boveri is chromosomes are called vehicles of heredity. That means they transfer the characters from the offspring's parents to the offsprings. Therefore, they are called vehicles of heredity. These chromosomes are responsible for carrying the information from previous generation to the offsprings. From their parents to the offsprings, the genetic information is being carried by the chromosomes. Therefore, they are called vehicles of heredity. They are transmitted from parents to offsprings. That is, they are immortal. That means they never have death. They do not die at all. Just simply they keep on carrying the characters from generation to generation. That is one of the important statements that is being made by the scientists. Two identical chromosomes with similar genes form a homologous pair. As we have studied about the homologous genes and heterologous pair will be there in the genes. That means if they are homozygous pair, they will be carrying identical similar type of genes. Next, homologous pair segregated during gamete formation. During the formation of the gametes, these homologous pair get segregated and they will be carrying the characters. That is another statement that is being made by them. And independent pairs segregate independently of each other. These homologous pair of chromosomes, when they segregate, they will be carrying the independent characters. They get separated, they get segregated and carries the independent characters. And chromosomes are mutable. They may undergo change, mutable. They can be changed. So chromosomes are mutable. These are some of the important statements that are being made from the chromosomal theory of inheritance. And genes are present on chromosomes, hence they show similar behaviors. These genes are present on the chromosomes. Millions of chromosomes, genes are present on the chromosomes. These chromosomes are responsible for carrying the characters. For example, one it is shown here, gene for eye color. If you look at the eyes of the every individuals, there may be a difference. It may be color, what we see. So that color of the eye is decided by one of the gene present on the chromosome. So uniqueness will be there. The beard shape, eyebrow, eye color, skin color, hair color, curly hair, long hair. These all are decided by the genes present on the chromosomes. Therefore, genes are present on the chromosomes, hence they show similar behaviors. These are some of these statements that are being made by the Sutton and Bowery, two scientists. And I repeat once again, they are chromosomes are vehicles of heredity. They are transmitted from parents to offsprings. That is, they are immortal. Second one, two identical chromosomes with similar genes form a homologous pair. Third one, homologous pair gets segregated during the gamete formation. Fourth one, independent pairs segregate independently of each other. And chromosomes are mutable. These are some of the important statements that are being made. And at last, genes are present on the chromosomes. As we all familiar, we know that genes are present on the chromosomes, hence they show similar behaviors. These are some of the important statements. Now, segregation of homologous chromosomes. You can see how the homologous chromosomes get separated. Very clearly it is shown that here you can see in this bivalent, it undergoes, the chromosome undergoes meiosis phase 1 anaphase, meiosis 2 phase anaphase, and in the germ cells these two stages will be occurring. Meiosis 1, meiosis 2 will be occurring in the reproductive part of the body, that is germ cells. That is, germ cells are being formed, and you can see how they got separated. So this is how segregation of homologous pairs. Homologous pairs of chromosomes get segregated, and they will be carrying the characters. This is how in this diagram also you can see both diagrams are same. You can see how the chromosomes are getting separated. The homologous chromosomes get separated, and independent assortment, they show the independency. You can see here same earlier what I shown, that same diagram is there. Meiosis 1 in the anaphase, meiosis 2 in the anaphase, meiosis 2 in the anaphase, where bivalent chromosomes are being formed, and they will be having the specific characters, and which will be carrying. At the germ cells, they get separated, they get segregated, and they become independent. That's how these will be there. And one more topic which was there is the sex determination in the animals. How the sex determination is there in the human being, we have studied already. Sex determination in the human being, we have studied, and earlier I have explained it. And now sex determination in the honeybee, how the sex is being determined in the honeybees, that we are going to study in this class. Now we can see honeybees, insect from which we extract the honey. What type of honeybees are there in the beehives? You can see male male honeybee is there, female honeybee is there, and also drone will be there. We call it as drone. In the previous classes we have studied up to the sex determination in the human being, and now let us see how the sex is being determined in the honeybee. Diploid female bee, little bit larger will be there in the size. It undergoes meiosis, producing the eggs. Here, when they produce the eggs, fertilization will not be there. Directly, if they produce the eggs, if fertilization is not there, then they will become the male haploid honeybees. They are called drones, what we say. And if you can say diploid female honeybee and haploid male bee, which is present, if they occur, this will produce the eggs and this will produce the sperm during the fertilization. When the fertilization takes place, then diploid female bees will be there. This is how it forms, and it is based on the number of sets of chromosomes and individual tissues. An offspring formed from the union of the sperm and egg develops as a female queen, or it is called worker bee. An unfertilized egg develops as a male, that is called drone. Here, these are the unfertilized. No fertilization is written here. You can see no fertilization. So, haploid male bees are there. In that, there is no fertilization. There is no fusion of sperms and eggs takes place. So, no fertilization will be there. They will become the drone, what we say. It is called parthenogenesis. The formation of the offsprings without the fertilization is called parthenogenesis, we say. So, this is one of the concepts that we have in the honeybees. Therefore, the females are diploid, 32 chromosomes signed. Males are haploid, 16 chromosomes are there. This is called haplodiploid sex determination system. In this system, males produce sperms by mitosis. They do not have father and thus cannot have sons, but have grandfather and can have grandsons. You can see here parents, females will be having 32 chromosomes in which they undergo the meiosis, produces 16 chromosomes each. During the formation of the gametes, they will be having 16 chromosomes, but male do not undergo any meiosis. Only mitotic cell division will be there, and therefore they will be having 16. During the fusion of male and female, they form 32. If they form 32, they will become female. And if they remain 16 only, then they are called the male. This is how the sex can be determined in the honeybees, one of the example. Another concept is mutation that we are going to study. Mutation is sudden, it is a, you can see how the mutation is there in the, it is a sudden heritable change occurring in the DNA sequences that results changes in the genotypic and phenotypic of an organism. The organism looks different. It's all because of the mutation undergoing changes in the DNA sequencing. When there is a changes in the DNA sequencing, then it is called the mutation. Recombination and mutation leads to variation in the DNA. As there is a variation in the DNA, results changes in the variation in the offsprings. These are the pictures where they have undergone the mutation. You can see if there is a mutation, this is what going to happen. So there are different types of mutation. One is frame shift mutation, another one is point mutation. There are types of mutations. One is frame shift mutation, means loss, deletion or gain, insertion, duplication of a DNA segment. If DNA segment is being lost, or if it is gained, insertion or duplicated, then it is called frame shift mutation. There also, it is also going to change. Then that is the change that is occurring is because frame shift mutation. Another type is point mutation. Mutation due to change in a single base pair of DNA. If there is, you can see in the diagram how the point mutation is being occurring. Single base pair is being changed and that leads to the mutation. That also leads to the changes. One of the example of the point mutation is sickle cell anemia. This is what it happens. Genes are located on chromosomes, so mutation results in abnormalities. Whenever there is mutation occurring, that results in the abnormalities. So abnormalities will be there because of the mutation. Chromosomal aberrations are seen in the cancer cells. These are seen in the cancer cells. Mutagens which brings the changes are called mutagens, we can say. Mutagens, agents inducing mutation, agents which are responsible for the mutation. There are some of the agents. One is called physical mutagens, physical agents. They are ultraviolet radiations, alpha rays, beta rays, gamma rays, X-rays are the physical agents that bring the mutation. Chemical agents are mustard gas, phenol, formalin, acetic acid, formic acid, NS3, ammonia are the chemical agents which are responsible for the mutation. So this is one of the concept called the mutation that we have studied. And another one, what we call as the P-degree analysis. P-degree analysis we will see in the next classes. Thank you.